help me welcome Dr. Giddings to Northern Vermont University.
Hi everyone, thank you so much for the invitation to come and speak with you guys about the exciting work that I'm doing with Middlebury students. So not down the street, but within the state of Vermont. I'm going to be talking about my work characterizing the microbes that live in acid rock drainage at an abandoned copper mine in Vermont, the Ely Copper Mine, which is actually in Orange County, which is south of here. And I'll be talking about the seasonal variations that we've seen in microbial populations, genes expressed, as well as our assessment on the environment's potential to produce novel secondary metabolites and transport metal. Before I get started, I do want to let you know more about who I am. So I am not a native Vermonter. I am originally from Brooklyn, New York. Is there anyone from New York here or New York State? Oh, cool, New York State? Okay, great. So I grew up in a city and I was fortunate enough to go to a science high school, a public high school that had a lot of really cool chemistry teachers. And I actually became interested in chemistry because I had about 3 different chemistry teachers who were all Black chemistry teachers, and they were very cool. And I liked science, and because we got along well and I looked up to them, I thought maybe I could do that too. So when I went to college, I actually went to an all-women's college in Massachusetts, Smith College. I decided to major in chemistry, and it was there that I started to get research experience. I also did summer research programs at the National Institutes of Health. So for some of you who are thinking about doing research during the summer, you can look for opportunities at not only your institution, but at other universities and government institutions. I would just apply because it gives you an idea of all of the different research that is going on outside of your school that you're unaware of. So it was me working at the National Institutes of Health that made me realize that I actually was interested in this field called natural products, which I'll tell you more about in a second. But I decided to go to graduate school to study natural products at MIT. And it was there that I also learned that I like to teach, and I took classes to learn how to teach science courses. And then I also mentored undergraduate students in the lab. And it was clear to me then that I wanted to teach, but teach at a place that I could also do research at. And so before you go and you apply for teaching jobs, you usually have to do a fellowship, so extra years of training. And so I went back to the National Institutes of Health, actually the National Cancer Institute in Maryland, and I worked trying to find new anti-cancer agents from microbes for about 3 years. And then I applied for academic jobs, and I ended up at Middlebury College. So that's kind of my life story. And one thing I do want to say, because it seems maybe that I had it mostly figured out, is that there was a point in my career where I didn't feel like I knew what I was doing. So when I went to MIT, I did a degree in I earned my Ph.D. in biological chemistry, but I actually started taking organic chemistry courses. I took organic chemistry and I took biochemistry courses, but I had never taken biochemistry before. So I had to learn everything that I know that I'm talking to you about today on my own. And I'm always learning, even now at Middlebury, doing such interdisciplinary work. And so it's very, it's normal for you not to really know what direction you're going in or your research is going in. But usually if you talk to enough people, you'll figure out the right path for you. With that, let me just get started. So, can everyone hear me? Can I talk like this and you can hear me in the back? Okay, perfect, because that was uncomfortable. Okay, so this is a really cool drawing. Actually, it's a painting that one of my undergraduate students made, and I think it really embodies what we're trying to accomplish here with looking at acid rock drainage environments. We're trying to look at these polluted environments. So they're metal-rich, acidic environments that microbes have somehow adapted to thrive in. Not all of them, but a subset of microbes. And we think that if you look at these polluted environments, that there are some benefits that we can find in these environments. So we can have these environments can potentially be sources of new chemical structures, new chemical diversity, bioactivity that could be useful for industrial purposes or for therapeutic purposes, as I'll talk a little bit more about in a second. So I am a natural products biochemist, and so I want to spend a bit of time explaining why natural products are so important before I tell you about our work at the acid rock drainage site at the Ely Copper Mine. And then I want to talk a little bit about some work that involves us not looking for new natural products, but trying to understand the enzymes that are involved in making them. So I'm going to talk to you about an enzyme that students and I have characterized that's involved in making a class of natural products called siderophores, and I'll end with that. So does anyone know any examples of natural products in the audience? Yeah.
Is it saying basically like it's organic, like it's fresh from the ground?
Yes, and usually they have some sort of biological target. Any ideas? Yeah.
I live in Tunbridge, Vermont, so the Ely mine is just a little ways away from my house, and I remember when I first moved in, reading in the local paper about them planting plants at the Ely Copper Mine to uptake the metals in the ground, and then they were taking from those plants, that harvested metal out of those plants, and taking it out and putting it back on the market.
Oh, that's interesting. I actually would like to talk to you more about that, because this is the first time I've heard about that, but that's probably some sort of remediation strategy for the site. Any other, so what about like any of the antibiotics that you may have heard of? Any idea? Pardon me? Penicillin. Penicillin. That is an example of an antibiotic that was found serendipitously, and it helps to kill off bacteria such as Staphylococcus aureus and so forth. So you have penicillin. Has anyone heard of artemisinin? Artemisinin. So there were scientists in 2015 that were they were awarded Nobel Prizes for their work with artemisinin, which is an anti-malarial compound produced by a plant, okay? And it's clinically used to treat malaria in different countries. So these are, in general, molecules, small molecules that are synthesized by a living organism. You'll see me use the word natural product or secondary metabolite interchangeably because we consider them to be the same thing. They're not required for the cell to live. They're thought to play a role in chemical defense. But these are small molecules that have distinct biological activities. So nature has evolved them to interact with specific biological targets. Now, they're called secondary metabolites, and why are they called secondary metabolites? Well, that's because secondary metabolites are these molecules that are produced by offshooting pathways from primary metabolic pathways. So primary metabolic pathways are pathways such as glycolysis, photosynthesis, the citric acid cycle. Some of you, have you guys heard of these pathways before? Yeah? So these are important pathways. They're primary metabolic pathways that produce these small, primary metabolites like glucose. These are molecules that are important for the cell to survive, okay? And the secondary metabolites, which I've kind of highlighted in these dotted boxes, they are these molecules that are produced by these offshooting pathways from primary metabolic pathways, but they're not important for the cell to grow and reproduce. They're thought to be produced as a means of chemical defense. And there are different classes and so forth. And I'll mention a few classes today, like siderophores. Natural products or secondary metabolites, they are structurally diverse. And we can just see from these different chemical structures how diverse they are. So artemisinin, what I had mentioned before, is this sort of terpene natural product that is produced by the Artemisia annua plant. It's an anti-parasitic agent. And the peroxy bridge right here is thought to be the active pharmacophore, or the component that gives it its anti-malarial activity. And then we have these N-methyldecahydroquinolines. These are these alkaloids that have alkynyl groups and alkenes, and they're important for, the activity of this compound or this class of compounds. And these are compounds that bind to nicotinic receptors and have some sort of effect on memory. And what's cool about them is these are molecules that are secreted from the skins of frogs. And then we have vancomycin, which is a last resort antibiotic, which is so different structurally from these smaller molecules. So we can have huge, molecules, cyclic molecules decorated with sugars. And this is a type of hybrid molecule of a non-ribosomal peptide and a polyketide linked all together. And it also has a unique halogen, like a chlorine atom. We don't typically see those in nature. And so, in any case, that chlorine and the sugars and so forth help to make this a very potent antibiotic. And it's produced by a soil bacterium, which is cool. So the structural diversity is really important for the function of these molecules. And the structure guides the bioactivity. And so it's not a surprise to then find that most of our FDA-approved small molecule drugs are inspired by natural products. Because remember, these compounds have been evolved by nature to selectively target something in the cell. So when we look at all of the FDA-approved drugs, what you'll notice is about a little more than a third of them are drugs that have been made purely synthetically, but then the rest of the drugs on the market are all somehow derived or inspired by natural products, okay? Which is very exciting. So, yeah.
Sorry, Sarah,
No.
isn't aspirin a version of this?
That's right,
So
salicylic acid. That's right. That's right. And I'm surprised nobody mentioned that before, but thank you for mentioning this. You guys know these molecules. You just didn't really put it together as a natural product. So it's then, important for us to find new natural products, not only because most of our drugs are inspired by these compounds, but these natural products help synthetic chemists come up with new methodology to access certain compounds, okay? So if you look at how nature puts these molecules together, that could inspire you to come up with a new chemical synthesis of a compound. And then you have some other uses for natural products. They could be used as biofuels if you make some of the carbohydrates or the terpenes or the lipids, you can burn them for fuel. A lot of, especially the terpenes, are perfumes, or they're found in perfumes. There's some sort of attractant or repellent. You'll find dyes and pigments are natural products. And certain flavors, again, the terpenes, your spices that you use when you cook, they're all different combinations of a class of natural products called terpenes. And natural products are also important for developing, for example, transgenic crops. Can we engineer crops that are drought resistant by manipulating some of the genes that are involved in a compound that could make the plant last longer under dry conditions? And we're also interested in engineering microorganisms like yeast and E. coli because, for example, with that Artemisinin compound that I mentioned, it is produced by a plant. And how long do you think it takes for a plant to grow? Yeah, a long time. E. coli has a doubling time of 20 minutes, whereas you have to wait and for a plant to grow, and who has time for that, right? So imagine if then you could find the genes that are involved in the biosynthesis of artemisinin, and you can then put them into a host like yeast, and you can grow a lot of cells, and you can access a lot of compounds. And this is what is happening already. This is how people are actually producing some of these drugs pretty cheaply. And that will also lower the cost of drugs. So it's very complicated to synthesize some of these molecules, but if you can put the genes that encode the enzymes that make these molecules into something like yeast and just have to wait and do an extraction for the compound, then that would make it, less time, you'd put less time into the process, and that could be a cheap alternative to producing these molecules. All right, so we're always looking for new natural products because, well, we're always trying to kind of fight our war with, for example, the antibiotic resistance that we're currently facing, and this problem is not going away. So the rate of resistance is rapidly increasing, and so we're always going to be in need of new antibiotics. We need to find new natural products. And of course, we're always in need of anti-cancer agents, drugs in general, in addition to biofuels. We're just looking for new chemistry. And it just so happens that we've exhausted all of our terrestrial sources that are easy to access for these molecules. Now we're finding the same things over and over in different organisms. And so now scientists are reevaluating their strategies, and they're trying to say, gee, where have we not looked? And so there are these extreme environments that took us a long time to develop the tools to access so that we could be able to study some of the microbes or the organisms that live there to see if they have any interest in metabolites. And so my lab focuses on studying, isolating microbes and their metabolites from unique extreme environments. So we have projects where we look at Antarctic microbes. So microbes that are from subglacial Antarctic ice. And then we also, as I'll talk about today, look at microbes that dwell in acid rock drainage, okay, at the Ely Copper Mine. And this is all work done in collaboration with a lot of people. So I collaborate with a subglacial microbial ecologist at the University of Tennessee, because that's the thing, you have natural products science research tends to be very interdisciplinary, because sometimes we need geologists to kind of assess the area that we're trying to study. We need microbiologists to help us figure out how to grow some of these organisms. We need molecular biologists to help us figure out the DNA. We need everybody. And so you'll see today how interdisciplinary, at least our work at the Ely Copper Mine is. So I work with Jill McCookie, and then with the Ely Copper Mine work, I've been in collaboration with a lot of people, a geologist, Holly Peterson at Guilford College, Heather Driscoll at Norwich University. She's a part of the Vermont Genetics Network, as well as a number of people at the University of Illinois at Chicago. And everybody has a different set of skills that they're contributing to the project. Some are bioinformaticians, some, like I said, are geologists, and so forth. Lastly, if I get to this project, hopefully I can, I'll mention that we're also interested in understanding the enzymes. So not only isolating microbes and new molecules, but we want to understand the enzymes that are involved in making some of these natural products, because we need to characterize them so that we can figure out how we can perhaps exploit them to improve the yields of certain natural products, okay? So we are working on a siderophore biosynthetic pathway that's found in microbes that live in soil, these soil actinomycetes. And this is work that I'm doing in collaboration with an X-ray crystallographer, because we're trying to crystallize some of these proteins, in addition to assaying them for activity and so forth. So we do a lot of different things in my lab. But let me tell you about our work at the Ely Copper Mine. So the Ely Copper Mine is one of 3 mines that make up the Vermont Copper Belt region. This is, again, in Orange County, Vermont, and I think is quite ironic that they're found in Orange County, because the soil in these metal-rich environments are orange from the iron that enriches these sites. But in any case, the Ely Copper Mine was intermittently mined from like the 1800s to the mid 1900s. And what's cool about this area is that at one point, Vermont was the leading producer of copper for the entire United States. Until they found copper ores in Michigan, and then that became the leading source of copper. And so it came from this region, which I think we're somewhere up here, right? Yeah? Yeah. Okay, we're down here. This is my first time in this area, so forgive me. But so, it's south of here, and what we wanted to do was we wanted to try to assess the microbial environment within the acid rock drainage at this site. And I'll tell you what that is in a second. And this was a seasonal study where we looked at collecting water and sediment at a particular brook, the Ely Brook, which actually washes it's a body of water that drains the mine, so it washes over all of the copper tailings, and because of that, it is acidic and it's metal rich. So we did a seasonal study where we collected samples in July of 2017 and January of 2018 to kind of see how the microbial population changes, gene expression changes, and again, to look for the potential of the microbes to produce new compounds, new secondary metabolites, and whether or not they have these genes that could be used to transport metal resistance genes. The reason we were interested in looking at that is because metal resistance genes could be used as a source of bioremediation. So if you can find genes that can chelate metal, you can use them as a means of developing filters or materials that can then be used to pull metal out of metal polluted environments, okay? So that's why we were interested in metal, and at some point we wanted to see if natural products and metals had some sort of relationship. So this will hopefully become clear soon. So what is acid rock drainage? Well, acid rock drainage is essentially something that forms when you have the metal sulfide enriched rocks at these mining sites exposed to air and water, the elements, okay? And what happens is the water and the oxygen get the water and the oxygen, they interact with the metal sulfides, and they, in addition to whatever ferric iron is around, will help to catalyze the oxidation of the sulfur, which makes up the sulfide and the metal sulfide. It becomes sulfate and H+, okay? Now, many of you, I'm not sure about your chemical knowledge, but if you have an abundance of H+, does anyone know what that means about the pH for the solution? It's acidic, because pH, by definition, is the negative log of the concentration of protons or hydronium ions. So if you have a lot of H+ around, you have an acidic solution or environment, which is what is happening in the Ely Brook at this mine. And the other thing about it is if you have any microbes around, they accelerate this process. They're probably one of the main drivers of this process, which does happen spontaneously, but they really accelerate the oxidation of the metal sulfide. So you generate a lot of acid and so forth. So we have these iron oxidizing chemolithoautotrophs that convert or help produce more of this sulfate and ferrous iron. Now, this is a picture of us working at the site over the last couple of years. So this is the brook, and we work at a certain location in the brook that, we've managed to collect water and sediment in the summer. So this was the summertime. Clearly, this is the winter time. And, it's kind of a situation where we're just making up, well, we're not making up what we're doing, but it's a kind of MacGyver situation where I come with a car battery, okay, which powers all of my pumps that I use to basically pump water through filters to capture microbes and DNA that we then use to sequence our environment, okay? So it's a pretty, I'm kind of hanging off. This is water here, and this is my feet, So I'm almost in the water, and on this day it was 2 degrees, okay, Fahrenheit, so it was cold. Okay, so our goal was to characterize the brook, the geophysical properties of the brook. So we knew what elements the microbes would have access to, and then we wanted to know what microbes live there and whether there were seasonal variations. We wanted to look at the genes that they express and whether there were seasonal variations there, because it also tells you about what the function of those genes are, within an environment, within a cold environment versus a very warm environment. And then we assessed the potential of the site to produce new secondary metabolites. We also looked for metal resistance genes, because remember, we're interested in them for bioremediation purposes, and we just wanted to assess what was present at the site. We also looked for antibiotic resistance genes. We did that because there is an idea, and it's actually been proven that wherever you have antibiotic resistance genes in a genome, and you have any genes that are involved in making these secondary metabolites, if they sit next to each other in a genome, chances are whatever that secondary metabolite is that sits next to the antibiotic resistance gene, it probably is an antibiotic. So they are actually working together. The antibiotic resistance gene becomes activated when the cell feels threatened, and to make sure that that microbe is not killed, the organism also has, this natural product, or the genes involved in making a natural product that'll be secreted to make sure that whatever was bothering it doesn't bother it anymore. All right, so that's why we wanted to look for antibiotic resistance genes. And then we wanted to know if there was any kind of connection between metal resistance and secondary metabolism. Just an idea. All right, so we had a whole host of methods used to carry out this work. First, we had to characterize the water, and so we went with a pH, or I should say a multi-parameter meter where we can measure pH, the conductivity, the redox potential of the environment. We also used mass spectrometry to determine what elements were present at the site, in the water, in the sediment. We also used X-ray fluorescence to learn more about the composition of the sediment and so forth. Here are some of our results. So what we have here are a number of different things. So you can look at the pH in general. It's about somewhere between 3 and 4 at this site. It's very acidic. Clearly, the water temperature is very different between the July, which we consider to be the summer, and January, which is the dead of winter. We did, these two extremes seasons for those differences. And this air temperature, the air temperature was also different, as one would imagine. What was interesting is that the levels of sulfate that we could measure were actually within the normal Environmental Protection Agency levels of tolerable sulfate levels. Anything above like 200 would be then a cause for concern. What we then also noticed is that the water had very limited levels of organic carbon, and that's normal at acid rock drainage sites because the acidic environment makes it hard for molecules to stay. It makes them unstable and they'll degrade. So obviously, we then expected there to not be that much carbon in the water. The redox potential told us, they're not that different, but it told us that the environment was mainly oxidized, and there was a difference in conductivity, and we do believe that perhaps there's a higher concentration of ions. It could be metal ions because we have more microbes in the summer that can actually produce more ferrous iron that accelerate that process that I had mentioned, going from the metal sulfide to dissolved ferrous iron. All right, so then we use mass spec to determine what elements were present and what we could really find. So this is the sediment data, and this is the water data. In general, we had higher concentrations of magnesium, iron. There was, copper, so, you would expect that to be there at the copper mine. But iron was really abundant in addition to aluminum. And then when we looked at the composition of the sediment, we also saw that we had a lot of aluminum oxides. There was silicon dioxide in addition to iron oxides and calcium oxide. So this is what was present in that community. Then what we did was we took those samples and we extracted the DNA and the RNA from them so that we can sequence them to see who is present, what microbes or what organisms live at this site. And we also wanted to look at the RNA, what genes are being expressed. If you want to look at the expression of a gene, you got to quantify the RNA transcripts, okay? And what we realized is that when we went and we collected, so we have water and sediment from 2 time points in the year, we noticed that all of our water samples that we took in the winter time actually didn't give us that much DNA. So we actually lost the data set, or we don't have data for the winter water. Because, it was very cold, so as you can imagine, we wouldn't want to live there. Who the heck, if you're a microbe, you wouldn't want to live there either. There'd be very few microbes in that water, and that's what we found. When we're talking about gene expression, we only focused on the genes that were expressed in sediment. Doing these experiments cost tens of thousands of dollars. So, when you're trying to figure out kind of how many replicates you need and samples you need, you have to think about, your budget. And so when it comes to the expression of genes, we focused on the genes in the sediment. All right. So in general, we extracted the DNA from our samples using kits, and we then created a library, a DNA library that we could use to do high throughput Illumina sequencing, where you can rapidly sequence entire genomes. And this is a metagenome now, because we're just scooping up soil or water that has lots of different microbes in it. It's not just a pure microbial isolate, So we're sequencing a metagenome. And so once we get the sequence data, what we do is we take those reads and then we try to annotate what taxa are present in those samples. So what kinds of organisms live there. So we did that. This is just showing a set of flowcharts, and then we split whatever taxa we found, we split them by superkingdoms. So we put all the archaea together, we put all the bacteria together, and since we're only interested really in microbes, we pulled out a kingdom, the fungal kingdom. So we're looking at fungi, bacteria, and archaea. And then we did some statistical analyses to kind of see what the diversity of microbes were in one sample, like in water samples. And then we looked at beta diversity, which will help us figure out what's the difference, or how diverse are sediment samples compared to water samples. So between samples, what are the differences in microbes as opposed to within one sample, which is what alpha diversity gets at. So we did those sorts of analyses and a differential analysis of the taxa. This is what we found. So before we actually just removed everything that wasn't a microbe, we plotted the data just to see what's present. The most striking thing to find for me was that we couldn't annotate most of the data. So everything was unknown. There are a lot of unknown rare taxa within this site. We could only annotate 25% of our sequence data, which shows that there's so many things there that we don't know. And when you think about assessing the potential for finding new chemical diversity, that's kind of a cool environment then to explore, because if we don't know anything about what grows there, then we definitely have chances of finding new molecules, because we don't know what those microbes are. So what we found, for what we could annotate, we had more bacteria. So we had bacteria that belonged to the phylum Proteobacteria. And then we did see, so when you're not thinking about microbes, but when you look at the entire group of organisms at the site, we did see chordata, which corresponds to vertebrates like fish and so forth, which you would expect in like an aqueous environment. which we found in the water. And so the fish would be shown in this kind of orangey color. This is bacteria. This is Actinobacteria. And so chordata can be fish and other animals, okay? And then we had some unclassified phyla, but for the most part, when we look at the site, microbes dominate the annotated sequences, all right? So mainly proteobacteria and then actinobacteria. And this is also exciting for a natural products biochemist because actinobacteria is this phylum that is known, or it has microbes that we know to be prolific producers of natural products. Okay, so the fact that a lot of them are here means that we have a chance of finding some natural products. Okay, so then we went on to do some statistical analyses where we look at the alpha diversity. So how diverse is this sample, this water sample, versus how diverse or different is the water sample from the sediment sample, which is the beta diversity. And what we found in general is that with the alpha diversity, so within a single sample, there were mainly the same things. So there wasn't a lot of diversity. The Shannon diversity indices were quite low for what we expect at all taxonomic levels. For the beta diversity, though, what we did see is that there were significant differences between summer bacteria in like summer water versus summer sediment. But what was also interesting is that between the seasons, so sediment from the summer and sediment from the winter, we didn't really see a difference. And we think that maybe we need to get more samples to be able to determine whether or not there was a difference. This is showing you kind of a representation of the main genera from different superkingdoms or domains. So we have archaea, bacteria, eukaryota. Also, we found viruses in this environment. We found a lot of different things. But when you look at this is a heat map that tells you about the abundance of different taxa in, for example, the summer. So this is the summer. You have the water samples here. You have sediment samples from the summer. And then you have sediment samples from the winter. And so you can see kind of that certain taxa only prefer to dwell in the sediment, and you can also sometimes see a difference between whether or not they're expressed or present in the summer versus winter, which is what this is showing you here. So there are some seasonal variations that are quite apparent. All right, I'm kind of going out of order here. Let me see. So what we then did was we wanted to look at gene expression, all right? So what genes are expressed and how do they vary with seasons? And to do this, it was a little bit more challenging. So what we did was we took our metagenomic data, which came from DNA, and we basically assembled all of the genomes together as best as we could. And then we went and we collected RNA from those same samples, and we sequenced them. So we ended up with a metatranscriptome. And for your RNA, what you do is you align your transcripts on top of the DNA so that you can then start to look at the expression of a certain gene. And so what we were able to do then, after we mapped RNA onto the DNA, is we were able to assemble genes and then predict their open reading frames. And we use a lot of different databases to then predict what genes were present in our data set. And I'm not going to go through a lot of this, but just know that we annotated the sequences, the genes, And then we did a lot of filtering to make sure that we didn't have any sort of false positives. So things with low counts in our data set were excluded and so forth. And this is a heat map of what we saw were the most expressed genes of what we could annotate. So again, when we looked at the genes that were expressed, the first thing we noticed is that most of the genes that are overexpressed in winter versus summer, we don't know what they are. They're all hypotheticals. Now, the ones that we actually could annotate is what I'm showing you here. So we have the same problem again like we had when we were kind of looking at the taxonomy, right? But on the gene level. But in any case, we were able to see seasonal variations, as you see in red with the sediment. So these genes here are really overexpressed in winter samples versus these genes here. And so we saw a significant variation in expression levels for certain genes. And the most abundant gene in the entire group of samples was this antibiotic resistance gene, which was also quite interesting to find. We could go in so many different directions with this. But what I do want to say is, of the genes that we could annotate, a lot of them had to do with amino acid metabolism or biosynthesis, cofactor biosynthesis, nucleotide biosynthesis, cell signaling. Basically, they all, these genes all have something to do with cells trying to live, to survive. So when a cell is stressed out, as you can imagine, it's trying to, make sure it has all of its key building blocks, like its lipids, its nucleotides. You need to make DNA. You need to replicate genes and so forth. And so these were all of the genes that were readily overexpressed. And the organisms that express them, most of them were unassigned. We don't know. So there's a lot to find here. What we then did was we looked to see if we could take our assembled genome, metagenome, and there's a website or a database, an online database, that curates all of the genes that are known to be involved in making a natural product, okay? So what we did was, using that database, we went and took our data and tried to find genes that could make some class of natural products. And so what we found from doing that was that we had 1,589 gene clusters, so different groups of genes that could make natural products, and 449 genes were actually expressed. So this is a point I want to make. Even though a gene may exist, or you may have DNA that could encode for some protein or whatever to be made, it doesn't mean that it's going to be made. The organism may have the ability to make it, but under the right conditions. And so what you find is, most of the time, microbes, they'll only, and humans, like, we only express certain things at certain times. If you don't need it, why would you, waste your resources to make everything you could possibly make? And so that's why only a subset of the genes or the gene clusters were expressed. Now, the types of molecules that we found, so we found DNA sequences or gene clusters that showed up as being 100% identical to known genes involved in making natural products such as geosmin, which gives what do you call it? I think it's mushrooms and earthy kind of flavor. Phomopsin, which is a microtubule polymerization inhibitor, so it's like a cytotoxic agent, and heptacine. And when we look at the different, so the gene clusters that would produce a specific type of natural product, the dominant type of natural product that we found was this class, non-ribosomal peptides, that the genes would be able to make, followed by terpenes, so a lot of terpenes. And terpenes are also used in cell signaling and so forth, which I don't know what their role is in that particular environment, but it's interesting that they're a dominant class. Okay. What we also did, because we had this expression data, is we wanted to know, well, which natural product gene clusters are expressed only in the summer or in the winter? Are there any seasonal variations with their expression? And what we can see from this graph, this is a dot plot of the expression. And here, so any dot that's at this level represents overexpression of a gene. And if it's in orange, it's a summer, it's a sample that's overexpressed in the summer, if it's up top here. And if it's blue, it's from the winter, or it's overexpressed in the winter. And you can see, so anything down here represents underexpression. So it's underexpressed. These are underexpressed in the winter, but overexpressed in the summer. What we deduce from this data is that when you look at the terpenes, it seems like most terpenes were overexpressed. Of the ones that were expressed, they were overexpressed in the summer, because there is a lot of orange here, okay? And then with the non-ribosomal peptides and any sort of hybrid molecule associated with non-ribosomal peptides, they also seem to be overexpressed in the summer. Why? I'm not sure. It's interesting though. Then we looked for metal resistance genes, and we found, so there's a database that has all of the known metal resistance genes, genes that have been experimentally validated as being metal resistant. And we annotated our data, and we were able to find 7,021 unique RNA transcripts that encode for metal resistance, all different types of metal resistance, copper resistance, cadmium resistance, zinc and iron, and so forth, which was interesting. And there appeared to be seasonal variations with that as well. Some are overexpressed in winter, but most of them tend to be overexpressed in the summer, which is interesting too, because we think that there are more metals in the summer, or metal ions in the summer, based on our conductivity values. The next thing that we did was we wanted to look at whether or not there was a connection between metal resistance and secondary metabolites. So what we found was weird, but cool. What we found were that there were some metal resistance genes that were sitting next to some genes involved in secondary metabolism. What that means is that this metal resistance gene, so for example, PIT-A, which is a gene that helps to transport phosphate and toxic metal ions in and out of a cell. This is sitting next to this kind of non-ribosomal peptide molecule. And we think that because they're next to each other, usually genes that are next to each other have something to do with each other, especially in microbial genomes. We think that perhaps this gene, if they're both expressed, they may be overexpressed together, because maybe in the summertime there's more phosphate or a toxic metal ion present, and then the microbe, in response to whatever happens to the cell when you're pumping out the phosphate or the metal, the toxic metal, ends up having an abundance of amino acids that can then be used to also make this secondary metabolite. Now, you know, what the biological activity is of this molecule, we don't know, but we kind of start to learn about the ecological roles of molecules, or why cells make them, by looking at, you know, some connections between like metal resistance genes sitting next to them. We also then looked at the antibiotic resistance genes using another online server that annotates known antibiotic resistance genes in your data set. And what we found were a number of different resistance genes. So this particular website looks for duplicated genes and genomes, because usually if you have duplicated kind of housekeeping genes, genes that are involved in some core essential function of the cell, usually that's a kind of, it suggests that the microbe is trying to overcome some resistance, so it gets several copies of the same gene in its genome. So that's one idea or way in which we get these antibiotic resistance genes. But then there are also just known resistance genes that don't necessarily require any sort of duplication. And so using what is in this database, we were able to find a lot of different known resistance models. There are a lot of duplicated housekeeping genes. And then what was really cool was that some of these genes, these antibiotic resistance genes, also sit next to secondary metabolic biosynthetic gene clusters. Now, what that means is that essentially, if you have some sort of secondary metabolic biosynthetic gene cluster here, and you have some sort of antibiotic resistant gene sitting nearby, chances are they're involved in the same thing. So if antibiotic resistance gene is activated because something is bothering the cell, and say, the biosynthetic gene cluster is next door, chances are it may also be activated, and it may be there to provide a molecule that makes sure that the cell is not going to die by whatever is troubling it, okay? And so what we then found was, so some metal resistance genes are also antibiotic resistance genes. So then we found a set of different metal resistance genes that were co-localized with genes that are involved in secondary metabolism, and they're co-expressed. So together, they're expressed in the summertime and underexpressed in the wintertime, and perhaps they're working together to make something to protect the cell. And so my thought is that we can use these metal polluted sites to find metal resistance genes that could be working with secondary metabolic biosynthetic gene clusters to make, for example, antibiotics, okay? Because again, a class of, or a subset of antibiotic resistance genes are metal resistant. And so perhaps in these metal-rich environments or metal-polluted environments, they're answers to some of our therapeutic problems, all right? So I'm just gonna briefly conclude by saying that most of the bacteria, so first of all, most of the organisms at the site were rare. We could not annotate them. There were mainly proteobacteria, which we could annotate. And then we found that there were a lot of unknown genes expressed, differentially expressed in summer versus winter. It was weird to find this multi-drug resistance gene being really overexpressed in data sets. And then we, again, found a number of different biosynthetic gene clusters that encode for terpenes and non-ribosomal peptides, and we were able to see differences in how these were expressed. And we found several resistance genes, antibiotic resistance genes, and metal resistance genes. And some metal resistance genes were co-localized and expressed with the biosynthetic gene clusters. So our thought moving forward, and we're trying to now use this as a hypothesis to do other work, is that perhaps metal resistance genes could be used to bioprospect metal-rich environments for new antibiotics. And in addition to that, just knowing where these resistance genes are next to these secondary metabolic gene clusters could be useful in understanding their ecological role in that environment. Now, I was not sure if I was going to have enough time to touch this Sedair-4 biosynthesis project. So I don't think I have enough time, but what I do want to say about it is that we also care about the enzymes, okay? We don't just care about the molecules, we care about the enzymes. And this project is a project that was done by students. It's continuously being done by students in a class that I teach, a biochemistry lab course that focuses on teaching students new biochemical techniques for the first 6 weeks, and then in the last 6 weeks, they put those skills to work. And what we were able to do as a class was find some genes that actually encode enzymes that had not been previously studied, that are involved in making this group of, or this class of natural products, but specifically this family of natural products, the desferrioxamine B set of molecules, which a Sedair-4 is a molecule that chelates metal, so the iron is not a part of the molecule, but everything else is the molecule, and they chelate metal, and they help to bring metal back to microbes that are living in nutrient-limiting environments. And this is actually a drug that's clinically used to treat metal poisoning. And what's cool is that pharmaceutical companies solely rely on fermentation to produce this. However, there have been supply issues, and this is why you want to understand how the biosynthetic pathway works and understand the enzymes so you can manipulate them to not have supply issues, okay? And that's why we started to work on this, and we've gotten really far. I will just have to come back again, or you'll have to read the paper that comes out on this. But it's some really cool work that we've done with undergraduates, and I'm not going to talk about it, but hopefully I'll have another opportunity to. A lot of people worked on these projects. A lot of my students have worked on these projects. The work that I talked about today was done by Catherine Murillo and Kieran Bahav. So Catherine, she was the girl with the mid hat that was on the snow or the ice or whatever. We were collecting samples. She did a lot of the DNA extractions, and Kieran did a lot of the analysis. As you can imagine, we got a lot of data. I mean, huge volumes of data. And so we had to sort through it to try to group and see any trends. And so Kieran was really instrumental in that. This is Kieran and this is Catherine. And there are a number of students that also helped with the biosynthetic gene or biosynthetic pathway that I didn't get to mention. But I do want to mention, again, my collaborators at the Ely Copper Mine, or at least with this project, the geologist Harley Peterson, all of these people at the University of Illinois at Chicago who do bioinformatics and sequencing, Kevin Kunzman, Stephen Green, Mark Manchen-Klein, George Chapala, also Heather Driscoll for her expertise with dealing with big data, and she helped me do a lot of cool plots and stuff. She's at Norwich University, and then I didn't get to talk about the second project, but my crystallographer, George Luntos. These funding sources, and you for your time. If you are interested in following us, I'm told by my group members that I should advertise our Instagram. We're on Twitter. We have a website, so please do check us out. And thank you so much for the invitation to come.
Thank you.
You guys have any questions? I know that was a lot. You can ask me about what it's like being from Brooklyn in Vermont. Feel free to ask away. It doesn't have to be a scientific question. Yeah.
Hi, my name's Elizabeth, and I was wondering about like the antibiotics, if you have any interest in the future of maybe finding the next new antibiotic.
Yes, I do, but I also know that I probably won't find one because when you're working at small institutions, the research goes really slow. I'd have to quit my job and go and work for like a company that, focuses on this and has, so many different samples. I mean, we're just looking at one particular site, one part of one site, Like the Ely mine has so many other locations, and we've just chosen this site because it was previously studied and it was noted to be quite acidic, and it's easy to access. We didn't go down into the shafts or anything like that. So, any other questions? Yeah.
About accessing the brook, did you have to go for anything legally, or could you just go down and take samples?
I had to notify who did I notify? Somebody at the Environmental Protection Agency. And at first we were escorted onto the site by a couple of their agents. The mine is operated by an independent company. However, they work with the EPA, or the EPA is always going to assess the land and so forth. So we asked for the owner's permission, but then we dealt with the EPA. And once they saw how not dangerous our sample site was, then we could go by ourselves. But they knew that, we were actively going there, and so you do have to let people know. Other questions? Yeah.
What would be then a function of like the overexpression of terpenes in the summer?
It could be, the other thing, what could it be? We were thinking of some way of communicating.
What is the normal function? Like, is there a major set of functions
for terpenes? I know, like with plants and so forth, that's a way of some plants to, like I said, it can deal with communication. But we're still trying to figure that out. We haven't kind of dived into what this really means to just study kind of the impact of this environment on terpenes produced. But something to do with signaling.
Another
Yeah.
comment, if there, my research, as but probably many of the students don't know, is looking at a similar metagenomic study, but in an alkaline environment. And that alkaline environment is the asbestos mine in Eden. And
Where's Eden? I'm sorry.
20
It's
minutes
north?
from, north from here, right?
Or a
Yeah.
little bit to the right.
Okay.
And many of our findings in terms of the sequencing data is very similar to yours.
Yeah.
Most of the sequences are not identifiable. We have antibiotic resistance genes, and we have heavy metal resistance genes, high levels of magnesium. So it's an alkaline environment, and I would be interested in comparing just the microbial organization.
Oh, yeah.
And we did only water analyses in the pit pump, but if students are interested, I think there's projects that you could do, just computer-based projects
Definitely.
that we might look at.
I am very interested in this.
Simply looking at comparative studies.
That's right. No, I'm totally open to, I mean, we have a wealth of information here. I don't know where it's going to take us, but we can go in so many different directions. I'm open to going anywhere. So yeah, for those of you who are interested in working with big data sets, and some of you, I don't know, do you guys have any computer science majors, or I'm not sure. Not yet? Oh, okay. But if you were interested in programming to help sort through data, I mean, this would be the type of project you'd want to work on, because it's not just looking at Excel, you know, or with like 2 columns in Excel. It's like looking at, I mean, I had like almost 1 million rows in an Excel spreadsheet with like, I don't know, 40 different columns, and it's just a lot of data. So that expertise would be useful if you are interested in coding.
Liz, did you say you have similar organisms in an alkaline environment that she's finding in an acidic environment?
Yes, based on classes, yes. But I would be interested in looking at the taxonomic data in greater detail to see how much similarity there is. But like the proteobacteria, we're seeing the Actinomyces. Basically, we're seeing some similarities there.
And most of your stuff were unknown, like you can classify
Most of it's
them.
unknown, exactly. Most of this DNA sequences, it might be the same, it's a very similar percentage, like you had 25% known. Our data is very similar to that.
And I don't know, like for your data, if you use, there's this new ARTS web server.
No, we did
You
not.
should check. Well, it only came out in the last 4 or 5 years, but if you're able to see if there are any resistance genes sitting next to a gene cluster, that would be really interesting to see if you could identify the function of some of these.
And we have not done that analysis.
Yeah.
One thing we do is,
The
in addition to
anti-smash.
the metagenomic analysis, we've cultured the organisms. So we have cultures to work with in terms of looking at gene expression in living organisms. Although, again, it's only probably 10% of the total.
That's right.
But we have organisms that are multi-drug resistant and heavy metal resistant.
Cool.
So some of this would be interesting to look at in situ as well.
I'm open to everything. Any other questions?
Out of curiosity, Sarah, how has it been living in little old Middlebury, Vermont, or the outward towns of Middlebury compared to New York City?
Well, let me tell you something. So I moved from Brooklyn to Western Massachusetts, which that was kind of like the first time I was in a wooded area like that. And then I moved to Boston, or the Cambridge area, which was then more city-like, but not as busy as New York. I love Cambridge. Then I moved to Maryland. I hated Western Maryland. It was not for me. It was a big city too, Frederick, Maryland. I didn't like it. When I moved to Middlebury, I loved it. I loved it. I met some people. I like nice things, and I like to drink nice things. And I moved into a place that was across the street from a sparkling wine bar. And we became best friends, me and the owner. And so I found people who were really interested in Vermont. And so I love Vermont. I was never sad to move here whatsoever. But if I want to go out and shop, then I'm going to New York or I'm going elsewhere, and that's fine, you know? But there's some people here that make a lot of cool things, a lot of nice whiskeys and so forth that, you know, I really like. And jewelry, and it's just, and people, I mean, I think Vermont is unique because you meet people who do things not for the money, but they do things because they genuinely like doing them. And I respect that. So anyway, thank you guys.
Thank you very much.